Hi, I'm Theresa Harbig from the University of Tübingen, and today I will present to you OmicsTIDE, a tool for the interactive exploration of trends in multi-omics data, and especially in transcriptomics and proteomics data. Transcriptome-proteome correlation is often not perfect. However, subgroups of genes might correlate well, while others correlate poorly. The central idea of our approach is to find and visualize subgroups with concordant or discordant behavior or trends across conditions and to analyze them functionally. When I talk about a concordant trend of a gene, I mean a trend that is the same in the transcriptome and in the proteome. For example, a trend that is continuously increasing in both datasets, which you can see here. In contrast, a discordant trend is a trend that is different in both datasets, for example, increasing in the transcriptome and decreasing in the proteome. Now we'll show you the principle of OmicsTIDE. As an input, we get two tables, one for the transcriptomic data and one for the proteomic data. The rows in the tables correspond to genes and columns correspond to conditions, such as time points, which need to be the same in both datasets. We concatenate these tables and conduct k-means clustering as if it was one dataset, which represents an early integration approach. As a result, each gene is placed into a cluster for the transcriptome and the proteome, and we can see if a gene follows the same or a different trend in both datasets. For example, gene 1 is placed in cluster 1 for both datasets and therefore shows concordant trends, while gene 2 is placed in cluster 2 in the transcriptomic dataset and cluster 3 in the proteomic dataset, which is a discordant behavior. We then visualize this data in our main visualization. Before clustering, the data has been z-score normalized to make it comparable across both datasets. The trends are visualized in centroid profile plots, where the centroid is visualized as a line and the bands correspond to the standard deviation. The trends are connected with a Sankey diagram, which allows us to analyze the intersections of genes in proteomic and transcriptomic trends. In contrast to most Sankey diagrams, the color gradient of the intersections is inverted. By doing this, we can immediately see with which other trends a trend intersects without having to follow the individual bands. I now want to demonstrate to you how OmicsTIDE works. We want to reproduce the results of a study from Hogendig et al. From 2019, and this is their teaser figure. They collected transcriptomic and proteomic data from 5 stages of neutrophil differentiation in humans. And they used dynamic transcriptome-proteome correlation networks to group the genes into modules. Modules contain a trend of the transcriptome and of the proteome, and therefore are related to the intersections in OmicsTIDE. The authors analyzed the functions of the modules with a mixture of GO terms and other systems. Here you can see some modules, which they annotated with biosynthesis and metabolism, Ross machinery, and specific renewal. As a first step, we want to see if we can visually identify the same modules in OmicsTIDE by analyzing the intersections. And visually, we could identify the same modules, but of course, we need to verify if the functional annotations are the same. Just because the modules look similar, it doesn't mean that the function is the same or that they contain the same genes. To compare the functional annotation, use another analysis of OmicsTIDE that can analyze one or multiple intersections in more detail, which you can see on the top visualization. Moreover, we can conduct GO-term enrichment for the 3 GO-term categories. The results are visualized in ranked bar charts, where the length of a bar corresponds to the negative logarithm of the p-value. We find that RNA-binding is the most significant term in molecular function, and therefore we can confirm the author's annotation. In a similar way, we can also confirm granule development. However, for the other 2 modules, it is not that easy. For example, Biosynthesis and metabolism is an extremely unspecific term, and many genes are annotated with related geo terms. We therefore want to compare the genes in concordant decreasing modules of OmicsTIDE and of the study manually. To do so, we need to combine the decreasing trends, which are the green and the violet trends. When we compare the sets of genes in concordant decreasing trends, we can find that there's a very high overlap between the results of OmicsTIDE and the study, and only very few genes are not in the intersection. We thereby can confirm biosynthesis and metabolism as well. However, the module Ross machinery could not be confirmed. There might be several reasons for this. For example, that the trends might be dominated by other genes, and we need to explore this further. For example, we could play with higher numbers of K. But all in all, we're coming to the same or similar conclusions as the authors, but our approach is much easier and much more automated, while the authors had to produce the results with a lot of manual analyses. In conclusion, OmicsTIDE offers an intuitive trend comparison, and in contrast to many other approaches, it is not a black box. Moreover, because of its simplicity, the approach can provide valuable starting points for other analyses. The usage of k-means allowed us a quick exploration using different k, but more advanced clustering algorithms will be implemented in the future. Also, we want to include other omics data, such as metabolomics data. Thank you for your attention, and I'm open for your questions.